This Jove protocol is about inferring long non-coding RNA protein interactions using biophysical approaches and in silico approaches. This particular work is in lieu of a pair of long non-coding RNA protein interactions which we have been working in our lab. So this is in relation to congenital pouch colon, a rare disease that we have been working here. And a couple of other long non-coding RNA protein interactions, we have also shown it using prostate cancer pairs as well. Although our work is a conjuncture of non-coding RNA protein interactions, this work would largely cover long non-coding RNA protein interactions.
Non-coding RNAs, as the name suggests, does not code for protein. Only 2% of human genome are coding. They can be classified on the basis of size as a small and long non-coding RNAs. Long non-coding RNAs, which were initially thought to be transcriptional noise or junk DNA, are now known to be involved in various regulatory mechanisms.
For deciphering the long non-coding RNA-protein interaction using in silico and biophysical approaches, first we need to get the variants. To achieve this, we ran the whole exome pipeline, which was benchmarked by our group, and it uses only open access tools. The details for the same are provided in the manuscript. Step 1 is to get raw reads in FASTQ format and do a quality check using a tool called FastQC. If the data passes the quality check, the reads are then aligned to a reference genome and we get the output as a SAM file.
The SAM files are then converted to BAM format and sorted for saving memory as well as to decrease the overall runtime with SAMTools import command followed by SAMTools sort and index. If multiple samples are subjected for analysis, MPileUp option with SAMTools is used. Variants can be called from the sorted BAM files using warscan at this point using MPileUp to SNP and MPileUp to indel commands. These commands can run with user-defined criterion as well. The sorted BAM file can then be converted to VCF format using SAMTools VCF tools commands, and then the VCF file can be subjected for further downstream analysis.
Long non-coding RNA quantification was performed with a two-step reaction process, that is through reverse transcription and real-time PCR. Reverse transcription was performed using Quantitech kit. The detailed protocol is described in the paper. Once the cDNA is ready, real-time PCR was performed using CyberGreen on Light Cycler 480 instrument. 3 independent experiments were performed with each sample run in triplicate. The expression levels of long non-coding RNAs were normalized with respect to housekeeping gap DH, and it was calculated using the delta-delta CT method. The detailed protocol is described in the manuscript.
For inferring interaction between two biomolecules, we have selected KIF13A, a 202 kilodalton trafficking protein and a bona fide gene causal to congenital pouch colon. The LncRNA non-HETSAT 002007 is known to play a role in regulation and is promiscuously interacting with KIF13A along with 5 other protein coding genes. The prediction of binding motifs in LncRNA and its interacting residues in KIF13A was carried out by extracting the FASTA file of LncRNA, and its secondary structure was predicted de novo based on the lowest energy and the number of binding sites present in the predicted models selected for best 2 fitted models. The prediction was carried out using SVM generated model by SciBlast search. For prediction of probable RNA binding sites of KIF13A was carried out using Pprint server. In order to investigate the interaction of LncRNA with KIF13A, we have first divided 1585 bases long LncRNA into 7 fragments according to the predicted secondary structure of LncRNA, which has been predicted with RNA-fold version 2 web server. The N-terminal and C-terminal regions of KIF13A protein were modeled with every fragment of LncRNA, and the top scoring modeled complex was generated using HDoc web server and refined by HEDOC server.
The 3D structures were generated for each fragment of the LncRNA were docked with the kinase motor domain R1 of using the HDoc server, and the minimum energy was predicted and its binding residues were also predicted. Furthermore, it was redefined using the Haddock server, wherein the similar positive and negative controls were also considered, and this has been analyzed using the individual energy, total energy score, and the negative z scores of the model complex. Finally, the effect of the mutation on the protein nucleic acid interaction was studied using support vector machine studies, in which five-fold cross-validation of the 86 non-homologous protein chains were considered with an accuracy of 75% and using maximum matches correlation coefficient value. And the study was developed wherein the long coding RNA and its interacting proteins are very fascinating to understand the regulatory aspects of the diseased phenotypes and mutations associated with it.
MicroScale thermophoresis. It's a biophysical technique which allows to measure strength of interactions between two biomolecules. It does this by measuring changes in fluorescence as a result of an IR-induced laser temperature change. The fluorescence detected can be intrinsic, which is from a tyrosine or a tryptophan molecule, or from a fluorophore attached to the target molecule. So, the basic principle of microscale thermophoresis involves two basic components. First is thermophoresis, which is a directed movement of molecules within a temperature gradient, resulting in change in the local concentration of the fluorescent molecule in the measured area. This movement depends on properties of the molecule like the size, charge, and the hydrogen shell changes. The second important component is TRIC, which is called temperature-related intensity change, which is an effect where the fluorescence intensity of the fluorophore itself is temperature dependent, which is strongly related to the chemical environment of the fluorophore. Now let's see how MST is measured. We perform the measurements in capillaries, and during the measurement, we monitor changes in the fluorescence of the labeled target mixed with different dilutions of unlabeled ligand molecule. So initially, we monitor the fluorescence of the sample before the IR laser is switched on to generate a baseline signal before any temperature change. After a few seconds, the induced temperature change by switching on the IR laser, you can see how the fluorescence intensity changes over time. This is the MST signal, and the difference between the bound and the unbound state defines the MST signal amplitude. Finally, to derive a KD, we set up a 12 or 16 point binding curve with each point measured in separate capillary. In each capillary, we measure the MST signal, and we then compare the normalized initial fluorescence FO to the fluorescence at the set point after heating F1. F1 is then divided by FO to get the F norm value, and this is plotted against the ligand concentration for that capillary. Similarly, is done for each capillary containing different concentration of ligand, and we fit this curve to derive a KD for this interaction.
We have shown here how an interaction between a non-long coding RNA and a protein is deciphered using in silico methods and validated using biophysical methods such as microscale thermophoresis. We have also shown here how such interactions could be involved in a rare disease like congenital pouch colon, and which we have been working on for many years.